Hello friends, myself Vibha Shukla. I am assistant professor in Axis Institute of Pharmacy. Today I am going to discuss microbiology. So what is microbiology? Microbiology is a study of biology of microorganisms. So first we have to know what are microorganisms. We know microorganisms are present everywhere around us. So what are microorganisms? Like the bacteria, algae, fungi, molds, and protozoa. Microorganisms are those which we cannot see from our naked eye. We have to use microscope to see the microorganisms. This field is concerned with the structure and function and classification of microorganisms. How they controlling their activities, how the microorganisms are used in industries, and what are the levels where microorganisms are used. What is history of microbiology? Basically माइक्रोबायोलॉजी कहाँ से शुरू हुई, कैसे आई, ये सब हम लोग माइक्रोबायोलॉजी की उसमें टॉपिक में जानते हैं. तो माइक्रोबायोलॉजी बेसिकली बिगिन विद द डेवलपमेंट ऑफ माइक्रो ऑर्गेनिजम्स. तो माइक्रो ऑर्गेनिजम्स जब आए, जब उनकी शुरुआत हुई, तभी हमारी माइक्रोबायोलॉजी स्टार्ट हुई. फर्स्ट एंटनी वन लीवेनहोक, ही इज अ ड्रैपर whose hobby was to discover a lens. So first he makes and decide to develop a microscope, because microscopes, when we use microscope, we have to use a lens. Without lens we cannot use microscope and we cannot see microorganisms. Here then Robert Koch, he was a German physician. He discovered that a specific microorganism is required for the specific disease. Means a certain microorganism is required to develop any disease. Then the microbiology was during 1800 to 1900 period. It's laid down for the development of microbiology. So various scientists came, given their theory, and microbiology is developing in 1800 to 1900 period. Then microorganisms, where we use microorganisms, are being used in industries, in various valuable products. Like जब हम लोग घर में भी when we make curd from milk, then overnight we prepare a milk for the preparation of curd. So where the lactobacillus, that is a microorganism that will grow in milk and produce a curd. So microorganisms is used in our daily life, and their microorganism have a different activities in living things also. Like there are viruses, there are bacteria, and they provide a simple vehicle in genetics technology also. And there are types of microorganisms. So in microorganisms, bacteria, fungi, yeast, protozoa, viruses are there. They are bacteria, this is a bacteria, this is a fungi, this is a protozoa, and this is a algae. So how scientists know which type of microorganism is there? They know by their structure. So microorganism, when we study any microorganism, we have to know the structure of that particular microorganism. तो माइक्रोबायोलॉजिस्ट जो थे वो स्ट्रक्चर के बेसिस पे माइक्रो ऑर्गेनिजम्स की स्टडी करते थे. देन overview of microorganism. Microorganisms are present everywhere, in everything which we cannot see from our naked eye. So we have to use a microscope to visualize the microorganisms. Here it is a microscope to see a different type of microorganisms, like bacteria, like fungi, like viruses. Then microorganisms. So these are the various structures of the microorganisms. By the structures we have to know this is a which type of microorganism. Like this Streptococcus pneumoniae, that causes pneumonia. So this is a type of structure that is Streptococcus pneumoniae have to cause pneumonia. Then this is a Clostridium tetani. This is bacteria responsible for causing a tetanus. So this is the structure of Clostridium tetani. Then there is Vibrio cholerae. So this is the structure of Vibrio cholerae, which is responsible for causing a cholera. So these are the various structures. By their structures we have to know which type of microorganism is this, and why this is responsible for causing any disease in human beings. There are branches of microbiology. So, on the basis of taxonomy, means on the basis of their names, we have to know which are the types of microbiology. So, first, there is a bacteriology. As name suggests, the bacteriology, it means the study of bacteria, that is called bacteriology. जिसमें हम लोग बैक्टीरिया की स्टडी करें, that is called bacteriology. Second one is immunology. Immunology means the study of immune system. So here we are studying the microorganisms. So we have to study about the immunity of microorganisms like bacteria, like viruses. And third one is mycology. Myco means fungi. So mycology, that consists study of fungi, study of yeast, mold, etc. Another one is nematology, the study of nematodes. Nematodes means roundworms. Roundworms are also a type of bacteria, but it's here a name specify that nematodes, it means the study of nematodes is nematology. Phycology, phyco means algae. So study of algae is called phycology. Next one is astromicrobiology. We have to know astrology, astronauts. Astrology means कि origin of life of microorganism on the earth. किसी भी चीज का इवेल्युएशन कैसे हुआ, एस्ट्रोनॉट्स जो होते हैं वो एस्ट्रोलॉजी के बेसिस पे किसी चीज के बारे में जानते हैं, तो दैट वी हैव टू स्टडी origin of life on the earth, that is called astromicrobiology. Second one is evolutionary microbiology. How the microorganisms evaluate on the earth. किस तरीके से microorganisms आए, कैसे हमने उनको जाना, that is called evolution. Third one is cellular microbiology. Cell. Cell is the structural and functional unit of life. All the living beings consist of cell. So here, when we study about the structure and function of any microbial cell, that is called cellular microbiology. Another one is systemic microbiology. Systemic means when anything is in a computational way. So computational and mathematical activities of any microorganisms, that is called systemic microbiology. Another one is microorganisms structure. So how we identify the bacteria, how we identify the yeast, how we identify the viruses, how we identify the fungi on the basis of their structures only. So when we prepare anything, any in industry level, when we're going to prepare anything and use bacteria, so when we use bacteria, there are different, this type of colonies have developed. So when these type of colonies have been developed, so microbiologists understand that these are the bacterial colonies. It means there is a bacteria that is developed in this preparation. Another one is microorganisms, bacteria. Bacteria are of two types, prokaryotic and eukaryotic. Prokaryotic is a very simple type of bacteria that contain cell wall, that contain nucleoids, but eukaryotes is composed of proper nuclear envelope that contains cytoplasmic reticulum, nucleolus. This is a multicellular, where prokaryote is unicellular. So on the basis of their structure, on the basis of their uh, lifestyle, we have to differentiate bacteria in prokaryotes and eukaryotes. Another, this is a difference between eukaryote and prokaryote on the basis of their characteristic. So as we already told you that definition, eukaryote is a clearly defined nuclear membrane, and prokaryote is a unicellular organism that contain a membrane bound organelle. There are very examples that eukaryote are animal, plant, fungi, and prokaryotes are bacteria and archaea. This is a type of microorganism. Cell size in eukaryote is a 10 to 100 micrometers, and in prokaryote is a less than 5 micrometer. So these are the various significant characteristic that defines eukaryote and prokaryotes. Another one is a bacteria type. It's another type of bacteria that is aerobic and anaerobic. Aerobic means requirement of oxygen, means any bacteria that require oxygen for their growth. Without oxygen, aerobic bacteria cannot grow. So These aerobic bacteria are not survive, they cannot survive in absence of oxygen. This example is Mycobacterium tuberculosis. We know tuberculosis, it's a very known disease, that is tuberculosis. So when any bacteria enters in any living things, and if the bacteria did not get oxygen there, it will not survive. So without oxygen, aerobic bacteria cannot survive in any living things. Whereas in anaerobic bacteria, they doesn't require oxygen to grow. So if they enter in any body, in any living things, and if they require to their growth, so they doesn't require oxygen there. So means without oxygen they can grow. There is an example of Clostridium tetani, like tetanus. We know about tetanus. So if there is a virus, if there is a bacteria like Clostridium tetani, it doesn't require oxygen. But there is a one specification: anaerobic bacteria can not use oxygen, but if oxygen is present anywhere, they can survive. But aerobic bacteria, if oxygen is present, not present anywhere, they will not survive and they will not grow. Aerobes and aerobes. Anaerobes, they doesn't require oxygen. They make lactic acid, they make ethanol. Aerobes, they use oxygen, they use a citric acid cycle. And here both means aerobic and anaerobic bacteria both makes ATP, that is adenosyl triphosphate. So this is a common parameter that aerobic and anaerobic both does. Next one is culture media. What is culture media? Culture media is a gel or liquid that contain nutrients which is used to grow bacteria or microorganisms. जैसे हम लोगों को अपनी लाइफ में सरवाइव करने के लिए कुछ मीडिया रिक्वायर होते हैं, अगर हम लोग को ऑक्सीजन ना मिले, वाटर ना मिले, फूड ना मिले, तो वी कैन नॉट सर्वाइव. सेम एज अगर बैक्टीरिया को, अगर माइक्रो ऑर्गेनिजम्स को कल्चर ना मिले, तो दे कैन नॉट सर्वाइव. So that is called a culture media, that media which provides to the microorganisms for their survival. This is also known as growth media because it's required by the microorganism for their growth. There are different types of culture media available for the growth of microorganisms. There are types of culture media based on their physical state: liquid media, solid media, semi-solid media, absence of and presence of oxygen, anaerobic media, aerobic media based on nutritional factor, simple media, synthetic media. So these all are the types of media which are required by the microorganisms for their survival and growth. So simple media includes only nutrient broth that contain meat extract and peptone water, which makes a nutrient broth for the survival of microorganisms. Next one is simple media preparation. How we prepare simple media? So simple media contain nutrient broth, that is meat extract, and nutrient agar that consists of peptone and NaCl. That all preparation consists of nutrient agar media, which is a simple media preparation. Next one is complex media. Complex means when we added two or more things in a one media, that is called complex media. So here we would use blood agar, we use nutrient ingredients, and when we are not able to identify the exact content, that is called complex media. Next, enriched media. That enriched means that is already developed. So where we use blood serum or egg for the medium for the survival of any microorganism. Next one is different media preparation. So when we use microorganisms in industries level, we have to use these media preparation. There are different type of methods like streak plate method, simple dilution method. By these techniques, we used different media preparation for the growth of microorganisms, for the growth of bacteria. Thank you.